For posters, the committee thought that we'd like to invite some people to give a 4-minute elevator-style presentation on their posters. And so we chose we chose 6 people. I'm going to read out their names. I would encourage you to come closer to the podium to give your talks. The 6 people are, sorry, I have to put my glasses on. Andy Chen, Peng, Keshav Singh, Nico Higgs, Philmo and Lassoued. And the idea of these elevator-style talks is that you get exactly 4 minutes. At the end of 4 minutes, I'll ask the people to turn off the PowerPoint and move to the next one. You get exactly 4 minutes to give this talk. And these, the talks that we chose were based entirely on abstract. And so all we did was said, we looked at these and said, what would be interesting to hear a 4-minute talk on? So this tells you that when you are writing an abstract next time, think about it if you want, because it's very important. The other thing that I will do in this is I won't give anything away about what's in the presentation. You have to do that in 4 minutes. I will read the title. So this should also tell you, next time you're writing a poster or making a scientific publication, think about the title. And so is Andy Chen somewhere? Is he? Okay, you're right there. Okay, so you're first up. Is the talks can go up before they start talking, but I will time you for exactly 4 minutes. When there's 1 minute left, I will hold up one finger, this finger, and then you'll know that you have 1 minute to finish. And then at the end of the 4 minutes, my alarm will go off. I'll ask the people in the audiovisual to switch to the next presentation. Okay, so are you ready? Andy's talk is entitled Application of Next-Gen Phenotyping Tools to Identify Genes Essential for the Survival of Winter Wheat. And if you come up, when you're ready, I'll start timing.
Okay, that's great. Okay, I'm ready.
Okay, go.
Do you remember the taste of your last birthday cake or the smell of freshly baked cookie coming out of the oven? I do. These memories are made possible because of wheat and are special because Canadian wheat flour is one of the best in the world. Canadian winter wheat, however, must endure a special challenge, and that's the Canadian winter. As most of us know, that Canadian winter is harsh, ruthless, and painfully long. Winter wheat plant can die due to winter-related stress, such as freezing temperature, dehydration, and frost heaving. Therefore, my project aims to identify the key genetic factors that is essential for in-field winter survival in Eastern Canada for winter wheat. And the reason why I emphasize Eastern Canada is because Eastern Canada produces more than 65% of the winter wheat in Canada. But of course, to do so, we must first phenotype our plant, and that's why we're all here today. Advancement in technology has changed the way plants are phenotyped. You know, images are captured in the ground or in the air via unmanned aerial vehicle, and these images have been applied to evaluate winter survival. In comparison to classical phenotyping such as visual estimation, the imagery-based methods serve as an alternative that is precise, objective, and potentially high throughput. Okay, where's the next? My hypothesis is that imagery-based method, whether ground or aerial, can allow for an objective characterization of in-field winter survival of winter wheat. And the data can be applied along with genomic data to identify key genetic factors that are essential for the in-field winter survival of winter wheat in Eastern Canada. Oh, going back, sorry. I was fortunate to work with a diversity panel of 450 wheat variety. This includes historic and recent winter wheat varieties, advanced breeding material, and also some winter hardy spring lines. Aerial and ground level imagery were captured at the beginning of May. This image was processed to produce readings that reflect the in-field survival of each individual plot. At the same time, we also genotyped our diversity panel with the 90K isolate SNP chip, which resulted in 20,000 polymorphic markers. The phenotypic data and the genotypic data would then combine to conduct a genome-wide association study to identify significant marker trait associations. In my results, 3 unique marker trait associations were detected across 3 testing environments. They were on chromosome 5A and 4A, and they accounted for from 5% to 10% of phenotypic variance. So that's very exciting for me to see. But our future direction is to identify candidate genes around significant markers using annotated sequence, a genome sequence of wheat, and to develop molecular markers to use marker-assisted breeding to develop suitable winter wheat varieties for Eastern Canada. Because I'm in Saskatchewan, so I have to say this, this part. What is that called? You know, today I talk a lot about improving genetics and stuff like that. But good genetics must be accompanied and be complemented by good management practice, such as planting on the right time in order to ensure an optimal winter survival for winter wheat. Thank you for listening.
The next talk is by, Peng, and it's entitled Improvement of Water Use Efficiency and Photosynthetic Performance Under Drought-Challenged Conditions by Targeting TOR Signaling.
Good afternoon, everyone. So in the following few minutes, I will talk about our progress on the photosynthesis and water use efficiency. This is belong to the PERC, Project 1.1. And these 2 traits are very important for agronomic breeding. Their functions recur the stomata, but in different directions. For example, photosynthesis requires the stomata opening and to take more CO2. But the water use efficiency, which is usually related to the drought resistance, requires the stomata closing for the preventing water loss. Our project will address this conflict and introduce TOR gene to improve both of these 2 important traits. And this key finding is the basic of our strategy. So this is the strategy this is our strategy, and I cannot talk too much about that, but I think if you are interested, the details are presented on the poster downstairs. So generally speaking, we introduce TOR gene into the rhodopsis and the wheat lands. And then we select the transgenic plant for the high photosynthetic performance, high water use efficiency, and high yield as the model plant. And then we use this model plant to establish a series of evaluation methods, and then apply this method to the wheat cultivar screening in the field condition. At the same time, we perform the high throughput transcriptome analysis to assist our research. And we believe this integrated method can help us dig out the molecular mechanism behind the phenotype. So I don't talk too much, but here I want to share a short time-lapse video to prove our model plant work well. And okay, let's start. We can see these 3 plants from the left to right is wild type and a transgenic plant from gut cell promoter-driven TOR and gut cell promoter-driven TOR kinase domain. And they start the drought treatment with the same amount of water, same amount of soil, and the soil was covered to avoid the water evaporation. And then it's about 2 weeks drought treatment. The phenotype is very clear here. And I think at this time point, it's exactly like this. We will start the recovery response, recovery assay to record the recovery response. And then we start it. You see, we reverted them, but the wild type is almost died. And the transgenic plant looks well and fully recovered, and they complete their reproductive growth. And I think this method proves our model plant work well, and we definitely will use this method to more crops. Okay, I think that's all we can talk today. And if you have any questions, you can find me in front of my poster downstairs in the following section. Thank you.
Boy, people are being very careful. That was 30 seconds wasted there. Okay, the next talk is by Keshav Singh. And it's entitled Utilization of UAV-based hyperspectral imaging technique on acquiring phenotypic characteristics of prairie crops. Ready to go?
Thank you. Good afternoon, everyone. So in my poster flash talk, I will cover broad overview of how we can utilize this high-resolution hyperspectral imagery for crops, wide range of crops in Saskatchewan. So my aim for this PERC project is determining the ideal wave bands, because if I can say what is hyperspectral is a combination of imaging and spectroscopy. So imaging is a two-dimensional thing, X and Y, and spectroscopy is which you can now look by the eyes above the range of RGB, that is a spectral scale. So if you can see this cube, this is defining everything. I can say is this, I can talk about always about the space and time. So here you can see the space is X and Y, and along the axis, which we cannot never see, is a spectral axis. So the space and which I'm doing the flying lot of time at every week, couple of times in a week, and that is time scale. So if this is a four-dimensional data, which is very complex to understand and get rid of a lot of non-linearity in the real time to get the best data set. So if I go further, yeah, so this is the, sorry. It's not playing so drone will move from the plot by plot and take the spectral image of each plot, and by using this spectral signature, which I will show you in the next slide, this is the main spectral feature along the Z axis, which I call the spectral axis, and each point in this space is defining the spectral signature. Like we have the fingerprints, so by matching all those fingerprints, how the thing is varying in the plant scale, how the crops is varying with the time, that we can retrieve by this information. So I have retrieved lot of, I compare lot of indices which are existing with my new indices, and so before that, why this radius region is important, I can say, because it's making a transition from visible to the near infrared region, and that is the transition phase from visible to the near infrared, which we cannot see, and most of the plants will show a vigorous behavior in that, because this is the transition phase. So in the next slide, this is the 32 basic wavelength I retrieve after running this crops model, and this is the 150 band out of the 32, which is, I can say, the important bands for the analysis and the getting out new indices. And there is a CPMI, that is so this is the CPMI is a canopy pod moisture index. I compare with the existing moisture index, and we are going to test this in the field, because I developed this in an indoor control environment before. So finally, this is a detection of the complex states, which are the subtle features which cannot be visible by the eyes, which are indicating the stress, disease response, water and nutrition relation, photosynthetic capacity like chlorophyll, and all what is the crop productivity in the real estate, like yield, biomass, all the things. Thank you, and I could not get the time to write the space to write my crew members, so I thankful to my supervisor, Professor Steve, and all the agronomy crew who helped me the guidance and helped me to count this data. Thank you.
Thank you. Everyone is cooperating with this time very well. We must have scared people, which is good. Okay, the next talk is by Nico Higgs, and it's deep learning for phenotyping in high-resolution images. And you're ready to go?
Yep. On the clock. All right, my name is Nico Higgs. I'm a research assistant at the University of Saskatchewan, working in 3.2 of the PERC project. So my supervisor is Professor Ian Stavness, who you saw present some of this work earlier today. And I'm going to be talking about deep plant phenomics, or DPP as we call it, which is a library that lets you leverage deep learning for phenotyping tasks. It was started by one of Ian's PhD students, Jordan Ubbins, who's also here today. The idea was that there's a lot of hype around deep learning and data science, and we want to see this in phenotyping, but we don't think it happens as much as people want it to. And so Jordan started this library to try and make it easier for people to use, and I've been working on it, trying to enhance it and add some more to it. Specifically, I worked on object detection and semantic segmentation, but it can also do regression and classification tasks as well. The images at the top there are some examples of object detection. We want to be able to find an object in an image, put a tight bounding box around it, and then even classify what that object is. At the bottom there are some examples of semantic segmentation, where we'll go pixel by pixel in an image to come up with a probability of what class it belongs to. And if we do that, in the middle there we have all the pixel probabilities, and we can then threshold that to generate a mask for whatever purpose you need to use it for. Something that comes up a lot with all of these is that we have really large images. There's a lot of hype with semantic segmentation, object detection in academia right now, and coming out of Google right now, but all of it's on these like benchmark data sets that are quite small. They're like 224 by 224 pixels. And when you read the paper in the literature, some of them will even try to say, they'll brag that, oh, our algorithm can scale up to 800 by 800, as if that's supposed to be impressive. But we have images that are like thousands of pixels along each dimension, and these off-the-shelf algorithms just don't work. They fail, and so we're trying to find ways to make use of that. And you can do things like you can split an image into little pieces, run the little pieces through, and then kind of collate at the end, but then you lose all the context of the original image, which is really where convolutional neural networks shine. And also sometimes if you take a random patch, you might cut a crop in half, you might cut out half a plant, and this can actually, these off-the-shelf algorithms will just fail. So we've had to implement our own custom ones to try and alleviate this. And long story short is they're working well. We're very happy with it. And we're working hard every day to try and make them more sophisticated and add more to them. With the little bit of time that I have here, I'm just going to go a little bit more in depth with how we do object detection. And so on the next slide here, oh, did I shut it right off? Does it need a... I'm not sure what I'm doing. Okay, there we go. So what we're trying to do is with that image in the top left, it's got some Carinata plants in it. We want to pass it through the neural network that you can kind of see laid out on the right there. And we want to get that second image on the left. We want to get those bounding boxes tightly around the plants, and we want to label them correctly as Carinata. Now, we don't actually get an image out of it. We actually just get a bunch of numbers. And I've tried to visualize that for you with that image in the bottom left there with the red and blue. What it's going to do, it's based loosely on the you only look once framework, which maybe some of you are familiar with. It's going to produce a tensor, S by S grid. Each grid spot's going to have a prediction of the confidence. So the red ones are low, the blue ones are high. It's going to have 4 numbers for the box. It's going to predict what class it is. We use techniques like non-maximal suppression to try and make sure we don't get too many boxes. And that's how it's going to output it as what we call a tensor, which is just like a 3D matrix or a multi-dimensional array. The other problem is that results are all on benchmark data sets made of cats and dogs. There's no plant ones. So we've had our own, but I've taken off-the-shelf ones, trained them with transfer learning on this data. Our customized ones are working better, which we're happy with. And there's a lot more I'd like to say, and if you want to hear more, you'll have to come find me by my poster.
All right, thank you. The next, oh, come on. The next talk is by Philmo Gu. You're there? Okay, good. And it's called Exploring 3D Data Stacks with Stereo Viewing and haptics.
Hello, my name is Philmo. I'm from University of Calgary, and I'll be presenting about the 3D data stacks, exploring 3D data stacks. So flowers are comprised of many beautiful geometric structures. However, much of their beauty is not observable. They are actually, they're only surface deep. Underneath the display of their florets, there lies a complex internal structure that supports the development and patterning of the flowers. So by using the technique called micro-CT, or microcomputed tomography, we can see the inside of a flower and in microscopic details and upload the scans onto a computer so that we can analyze them. And one such analysis that we do is the analysis of structurally important components, such as the vasculatures of a plant. So there are a lot of ways to segment, and how we segment is by using computer vision. But there is no such thing as a perfect segmentation algorithm. As a result, what we do is we include humans into the segmentation process to increase the accuracy of the algorithm. So to do that, we made two tools. So one of them is called Shiver, or also known as Segmentation and Haptic of Volumetric Rendering, and Vine, which is called the Virtual Network Explorer. So Shiver aids segmentation by combining the automatic and interactive techniques of segmentation, such as haptics, or tactile feedback, so that the user can feel and be guided by the structure, and also stereoscopic display, so that the user can see the data as a 3D object. Vine, on the other hand, aids the user by immersing them in immersive 3D virtual environment. In the poster section, we have set up demos so that the users, guests, can try out our software, and we hope we can see you there. Thank you.
Okay, thank you. And now we come to the last of the elevator talks. And this is by, I hope I'm pronouncing this right, Lassoued. Or Reem Lassoued. And it's called Risks and Benefits of New Breeding Techniques.
Hi all. Reem Lassoued, Research Associate at the Department of Agriculture. Reem Lassoued, Research Associate at the Department of Agriculture and Resource Economics, University of Saskatchewan. We are all aware that climate change, population growth, and the rising demand for energy, water, and land are battling our world. We also know that these challenges are also affecting global food security. The key question, how can we feed a future 9 billion equitably, healthily, and sustainably? Science and technology provide practical solutions to tackle these problems. One of these solutions is precision breeding. Scientific community agrees that the application of new breeding techniques such as gene editing is an effective and timely solution that can help address the rising food demand for food via sustainable intensification of agriculture. However, the development and the commercialization of NBT-derived crops depend on their regulatory status and social acceptance. Over the last 3 years, my team and I have recruited over 100 experts around the globe to investigate their opinions on the application of new breeding techniques. Our panel includes scientific experts, government officials, and agribusiness professionals. Half of our panel reside in North America and a quarter in Europe. We asked the panelists a number of topics: technological uncertainty around new breeding techniques, regulatory social barriers, their cost from discovery to commercialization, risks, benefits. My poster mainly focuses on the risks and benefits of NBT-derived crops. Before moving quickly to the results, anyone already participated in one of these surveys? Well, it's not too late. Some of these are available on our project website. So results from the surveys on the benefits and risks. One key finding is that most of our experts believe that gene editing crops can generate more benefits than GM and conventional crops. A list of these benefits can be found on my poster number 51. Experts also indicate that these new crops present zero to slight risk to society on the economy, environment, and human health. Another key finding is that national regulation for health and safety, export market, consumers, and the media play a major role in determining how and where new breeding techniques will be used and developed in agriculture. Last but not least, most of our experts agree that food security, impacts on biodiversity and conservation, and compliance with food safety measures are very important socioeconomic considerations that should be included in national policy governing agriculture. Our ongoing survey investigates whether and how current agricultural policy should be amended to accommodate existing and potentially future new breeding techniques and derived products. Thank you, and I will be more than happy to answer any question related to my projects.
Okay, thanks everyone for participating. I hope that it's my hope that these elevator-style talks have persuaded some of you that it's a good idea to go look at posters. I was asked to mention a couple of things at this point. One is that all of the abstracts of the posters are available in the app. So if you download the app, then you can see the poster, all the poster abstracts there, and decide which ones you would like to see first. The other thing that I've been asked to point out is that there is one and only one way to vote for the People's Choice Award, and that is with the app. So if you want to vote for the People's Choice poster, you need to download that app, and I hope you enjoy the posters as much as we did reading the abstracts and thinking about them. Thank you.